So good morning, everybody. Thank you for coming. Thanks to the organizers for inviting me. If I could have on-screen presentation. I'm not on screen. There we go. Not full screen, but that's okay. I'm going to just start off before I shift into my talk with a shameless plug while you can watch this nice video over here. And the shameless plug is, the organizers will excuse me for the slight correction, but both in the introduction and in the pamphlet, I'm introduced as the head of the Department of Neurobiology at Weitzman Institute, and thankfully that's no longer true. We're in fact 2 chairs beyond me. But sadly, what I still am is director of the Israeli National Institute of Brain Imaging. And the reason I'm taking advantage of this plug here is because we have in the audience a lot of members of the Israeli brain imaging community. So I want to take this opportunity to share with you, as you can see in the time-lapsed movie showing here at the bottom, that we have now installed at the Weitzman Institute a 7 Tesla MRI machine, the first one in the Middle East, and in fact the first one of its kind in the world. It's a 32-channel PTX machine. So this is really state-of-the-art human brain neuroimaging, and the reason I'm bringing it up is because we are open for business, and what I mean for that is that we're a national center, and we want everybody to come and scan on this machine. It's a unique opportunity, and moreover, currently we have very, very generous support mechanisms to enable scientists and clinicians from throughout Israel to use the scanner. So really at a rate probably lower than your local lower power scanner. So this is a real opportunity, and I urge you to take advantage of it. Now I'll fast forward to the end of this time-lapse movie just so you see how things look. That's actually our center. That's not an illustration, and it's waiting for you to come. So with that behind us, I will shift to today's program, and hopefully, there we go. So many of you may be somewhat familiar with our lab's work regarding its primary theme, which is study of the human olfactory system. And here you indeed see a happy participant in one of our experiments, where what we do here is we take advantage of a very unique opportunity that the olfactory system provides us, and that is that its sensors, which are bipolar sensory neurons, these are actually a part of your CNS, are in fact out of the skull, right? So bipolar sensory neurons that sense odorants are placed right here at the cribriform plate, and we have a set of devices, including this is an odor source that generates odors into this area with 2 millisecond onset resolution. And more critically is what you see here is an electrode that travels up right about to here and can record directly from these neurons in a behaving human, which is a rather unique opportunity, and also can microstimulate these neurons to impact brain activity. And this world of studying the olfactory system is in fact most of what we've done for the past 20 years. But recently this has taken a tangent, a related tangent, and I'm here to tell you today about that tangent. So if you were to go into the Oxford Dictionary and enter the term inspiration, you would receive 2 types of definition for this word. One is simply the drawing in of breath, that action, that's inspiration. Another type of definition you would receive relates to mental processes. For example, the process of being mentally stimulated, a sudden brilliant, creative, or timely idea. And we were wondering, why are these mental processes related to one respiratory phase and not the other? In other words, why is an idea not expiring, right? Why didn't I have a great expiring idea? Why did I have a great inspiring idea? What's this link between respiratory phase and mentation? And we have a potential answer to this big question that's olfaction inspired, if I may use that word. So what's our olfaction-inspired theory? If you think of the olfactory process, stimulus acquisition is inherently linked to the act of inhalation. What you see here is a composite fMRI of the brains of a bunch of people, and what you see is activation from the act of sniffing alone, without any odorant embedded in the odor. And this is just a re-demonstration of what was known since the work of Adrian in the early 40s in the hedgehog, that just sniffing, just inspiration, is in fact the greatest activator of the olfactory system. Because if you think of it again in olfaction, the stimulus is linked in a one-to-one relationship with the act of inhalation. You have to inhale in order to obtain the odorant. You have to do that in order for the odorant to enter your system. And therefore, these two actions are linked, inseparable. And this has dominated the neural organization of this system. In other words, sniffing alone drives the volley of neural activity throughout the entire brain olfactory systems in anticipation for the stimulus. Okay, so there's this stimulus processing phase that's linked to inhalation. Now, olfaction happens to be the most ancient mammalian sensory system, and there are many lines of evidence implying that it in fact guided the evolution of the mammalian brain. And our hypothesis is that this element remained throughout cognitive processing, and in fact, just as inhalation drives a stimulus processing mode in olfaction, we hypothesize that inhalation may drive a stimulus processing mode throughout the brain, no matter what the task. In other words, we hypothesized an inhaling brain. Now, we set out to test this hypothesis in a bunch of experiments, which I'll briefly tell you about. And primarily what we did is we measured people's respiration, initially using a nasal cannula, it's this device you see here, and had them perform an assortment of cognitive tasks completely unrelated to olfaction. So for example, they performed a visuospatial task, which I'll talk about a lot today, which is just deciding if a particular shape on the screen can or cannot exist in the real world. They did all sorts of math tasks where they had to decide if the solution was correct or incorrect, and verbal or lexical decision tasks, for example, deciding if this is a word or a non-word. And the way the first set of experiments worked was that participants sat in front of the monitor, and their instructions were to press the button the minute they felt ready to do the task as fast and as accurately as possible. Now, what we could then do is we could look at their button presses and see how they aligned with the respiratory trace. Okay? And what we find is something very straightforward, and that is that throughout these tasks, people inhale when they opt to do the cognitive task. Now, these are completely non-olfactory cognitive tasks, yes? But they opt to inhale, and I'm at an angle here, so it's a bit hard for me to use the laser pointer, but basically what you see in black is the traces of all these inhalations. These are 30 participants on this graph, and in blue is the average, and the dotted line at time 0 is when they press the button. And you can see that they pressed the button and immediately inhaled. The orange line is showing you the statistical significance of this, and it's highly significant. And of course, we compared this to random data and compared this to buttons press alone and so on and so forth. People tend to inhale when they do a cognitive task. Okay, so we've seen this natural tendency now, but does it have any functional meaning? So in a second task, what we did now is we provided our participants green? Yeah, way better. So we provided our participants, in the second set of tasks, participants did not self-initiate the task. They did not press a button to do the task. But what we did is we recorded their nasal respiration unbeknownst to them, and we triggered the task according to their respiratory trace, okay? So participants can get a trial either during inhale or during exhale, and these trials were interspersed throughout a long experiment, and this triggering mechanism was not known to them. And we observed two phenomena. First, we find that the brain processes this exact same task very differently if encountered on inhale or exhale. And I'll concentrate, for example, just here, and what you have here is an EEG recording, and you have the evoked response in the negative 200 millisecond zone, and you can see in orange the response while doing the task in the inhale phase, and in blue the response while doing the task in the exhale phase. And you see this pronounced and very significant difference here, that is, the brain processes the exact same task differently when an inhale or an exhale, and this is in non-olfactory regions. Now, finally, and in fact, in a way most critically, so we see not only do people opt to inhale at a non-cognitive task, not only does inhalation drive different brain activity than exhalation, but arguably most critically, performance is dramatically affected by this. And I'll grab your attention to the graph on the right, and what you see here is all the participants, and both axes are percent accuracy, and you have on the x-axis percent accuracy in inhale, on the y-axis percent accuracy in exhale. This is the unit slope line, right, where x equals y. So if people were equally good when doing a task at inhale or exhale, the points would be distributed around the line. But no, you see all the points, all 30 participants are in fact, or almost all 30 participants are in fact better in inhale over exhale. And here you can see this graphed as in the way people may be more used to seeing these types of things. And you can see performance in inhale and performance in exhale. People are better. Okay? So what I've shown you is that there's a very strong link between non-olfactory cognition and nasal airflow. However, as they say in American advertisements, but wait, there's more. Okay? So I've shown you a link between nasal inhalation and cognition. And now I'll invite all of you in the room to do a small, simple experiment. Occlude one nostril, not by putting your finger in your nostril, but rather by pressing on it from the side like this, and inhale. And then occlude the other nostril and inhale. How many people in the room felt a pronounced difference in airflow between the nostrils? One is easier than the other. Okay, an overwhelming majority of the room felt this difference. Fear not, we've not just diagnosed you with some horrible disease. In fact, what this is a manifestation of a phenomenon known as the nasal cycle. Okay, your airflow shifts between dominantly left nostril and dominantly right nostril every 2.5 hours or so. Okay, so you have this ongoing shift in nasal airflow. Now, remember, we said that nasal airflow impacts brain activity. We showed that both in recording and in behavior. Therefore, the impact of the nasal cycle is that you also have asymmetric brain activity as a function of this asymmetric flow. Okay, I hope everybody's with me. I took you through a certain path here, but you have this asymmetric flow and asymmetric brain activity. Now, where did this come from? What's going on? Okay, so each one of us in our nose, and this is a coronal image of the nasal passage, we have these structures that you see here and here. These structures are called the turbinate. This is in fact the inferior turbinate. These structures engorge with blood flow asymmetrically, physically blocking either one nostril or the other. So the reason we have this difference in airflow is because one nostril is physically blocked. Now, this asymmetry in blood flow is the reflection of asymmetry in our autonomic nervous system. Okay, when we have high, this is a slight simplification, but when we have high sympathetic tone, then we will have higher flow through our right nostril, and we have higher parasympathetic tone, we will have higher blood flow through our left nostril. And what you see here is a demonstration where we pharmacologically change parasympathetic and sympathetic tone and have this alteration between left and right nostril flow. And here you can in fact see the MRI of the nasal passage opening and closing over time, but I won't go into the details of this. So we have this cycle in our nose. This cycle is reflected in nasal airflow, but in fact reflects the level of balance in our autonomic nervous system, right? If we could get control over this, if we could control our nasal cycle, we could relax, right? Because it's well known that ideal balance between sympathetic and parasympathetic tone puts you in a rather relaxed state. In fact, this has been well known to Indian medicine for about 2,000 years, and they have a whole world surrounding this, but it's been significantly overlooked by Western medicine. Okay, so we have a key to relaxation. And of course, the way to always succeed in a presentation is show a kitten It always works. Now, is there any reason to think that we could be able to get a control over our nasal cycle, that we could control our nasal cycle through the power of thought, if you were? To test this, what we did is we gave people cognitive tasks that are known as either right hemisphere drivers or left hemisphere drivers. So these are either spatial tasks or verbal tasks. And here you see the results from 30 individuals performing 8 some tasks. And you can see that they start off from an equated point. I mean, we equate the point of start off, and you can see that as you do verbal tasks, then left opens, and as you do spatial tasks, right opens, or in other words, you can control your nasal cycle with the power of thought alone. This brings us to our final slide and to the project that was graciously funded by Joy Ventures and that brings me here today. And that is that we set out to do exactly this. So we developed a wearable device. It's called the mustache. You can see it here, you can see it here, and you can see it here. I think from you standing here, that means I don't have much time left to actually demonstrate it, but it actually transmits by Bluetooth, has onboard memory, has a host of functions that it can perform. I'm in fact wearing the mustache right now, and it's actually right now transmitting. And with the biofeedback software, we're teaching people to gain control over their own nasal cycle in order to generate a balance in the autonomic nervous system and relax. So you can learn how to increase flow through your left nostril, drive increased left hemisphere brain activity, and increased relaxation. With that, I would like to first thank our various funders in our lab that are primarily the European Union. Without the European Union, we would be dead in the water. But this particular project was funded by Joy Ventures up till now. And key players in this project were the engineers Aaron Weisbrod and Daniel Honigstein, and Ofer Pearl, who ran the experiments you saw. Of course, I'd like to thank all our group. Thank you for your attention. And I'll be happy to take any questions that you may have. Thank you.